Hello, everybody, and welcome to this episode of Genomics Lite. My name is Sam.
And I'm Jack. We both work in the Wellcome Connecting Science team on the Wellcome Genome campus. If you haven't joined us before, then Genomics Lite is a series of short sessions that share the science that happens here on our campus, which is all about DNA codes. This is our first video of our autumn-winter series, exploring some key questions we get from people like you.
And the series is streaming live on YouTube. So welcome if you are joining us right now on the live stream. And all of our genomics slides will be available afterwards on our YouTube channel. So after you finish this one, please do go and check out some of our other videos. We ran a whole series over the summer exploring lots of different and interesting concepts to do with DNA science. We also have released just last week some really short 1-minute clips. So if you're in a rush and you need your DNA fix, those are the places to go. So do go and check those out, but don't go anywhere just yet, because this session is all about exploring how DNA mutates. And we're going to be delving into that. We're going to be looking at what we mean when we say the word mutation, how this occurs, and what it might mean in terms of potentially, risks of disease and stuff like that. And we will be joined by two amazing guests, and we'll introduce those very shortly. But first of all, Jack, what do we need to know about DNA? Well, get us back on the same page. We've had summer, get us back to the starting point. What do we need to know about DNA and this word mutation?
Yeah, absolutely. So first off, when we talk about DNA, we're talking about a code. And like all codes, it's made up of specific building blocks. For DNA, we call these DNA bases. They are 4 different chemicals, but when we work with them on our site, give them 4 letters, and they're just the first letter of the name of each of those chemicals. So it's A, C, T, and G. Now, in the human genome, that word for the whole DNA code, so in the human DNA code, we have 3.2 billion letters, these building blocks that make up that DNA code. But sometimes those letters can change. And when they do, when those DNA bases change, the letters, we call this a mutation. Now, why is this important? Well, the order of those letters in the DNA code gives our body instructions. And so if it's a different set of letters, or the order is mixed up in some way by those mutations, then that's going to cause some kind of difference in the instructions. Now, we also need to think about what kind of scale mutations happen on. You can have really small scale mutations where it just affects maybe 1 or 2 letters, those DNA bases. Or you can have much larger scale mutations where a whole section of DNA code is changed, removed, added, swapped around, all sorts of things. So different scales for mutations as well.
And often when we hear this word mutation, potentially, in sort of popular culture, science fiction as well, mutation feels quite a maybe scary, negative, maybe unnatural word a little bit. It certainly potentially has those, sometimes comes to mind when I sort of see that word, but it isn't quite like that, is it?
No, that's right. Mutations can and do happen throughout our body, throughout our lifetime. We're going to be delving into this more with our guests, but there are lots of mutations in your DNA that have no effect whatsoever that we can see. These are what we'd call silent mutations. And the vast majority of mutations that we have in our DNA codes are silent. They don't have an impact. In some circumstances, mutations can be inherited, they're passed down, and these can contribute to that genetic variation that we call evolution, the way that species change. So there we see a really positive effect, species changing as they adapt to their surroundings all the time. And that's when that mutation, that random change in their DNA, happens to have a positive effect on their likelihood to survive and to breed, to pass on those genes. There are, though, and you said normally they have negative connotations in popular culture, and that has some good founding, because there are mutations that can cause us problems. They can cause us diseases, and the most famous of these that affects the most people is cancer. That's a suite of loads of different conditions that are caused by changes in our DNA. One example of that is we see a mutation in one of the genes, a section of our DNA code, where for a gene called the BRAF gene. And this is a gene that helps control how our cells grow. And a mutation to that particular gene can make those cells grow exponentially, and that becomes a cancerous tumor.
And I think you started to touch upon a little bit, but how do these mutations start to come about? Again, this is something we'll clearly delve into with our guests. But
yeah. This is the main topic of this episode, so I'm not going to give the game away too soon. But yeah, there are a couple of different. I'll give a couple of examples where we can see DNA changes that are mutations. So the first thing to say is that our cells have to make copies of our DNA and use our DNA all the time. It's not just a book that's sat on a shelf and it's completely stable, doesn't get touched. Whenever our cells interact within that DNA, there's opportunity for mistakes to happen, for breaks to happen, for all sorts of things to happen to that DNA code. Now, normally our cells are pretty good at spotting this and at repairing any damage. But if they don't repair that damage, then that becomes what we call a fixed mutation, a mutation which is there in the DNA code. Now, I mentioned there that the DNA needs to be copied. This happens when we make new cells, and we're making new cells all the time. Your body will be making new cells right now. As it does that, it needs to take the DNA, split it apart, and make 2 copies out of the same bit of DNA code. It's really good at that, but it's making copies of 3.2 billion letters. And so naturally, there will be a few mistakes in that DNA replication, as it's called, that photocopying of the DNA code. It's a natural process, and that's one way that we can get mutations into our cells. Another way is if the DNA is damaged in some way, and you'll have heard of this, when we talk about the problems that people have from smoking, or if you don't put sun cream on, UV light on your skin, radiation from a nuclear explosion, if we're talking about a sci-fi film or something like that. All of these can be ways that our DNA can be damaged, and those damages might become mutations.
Okay, perfect. So I think we've started to unpick this, but hopefully what comes across to our viewers is, and hopefully what we'll now start diving in, is that there's lots of nuances to this, and hopefully we'll start to unpick some of this with our guests. So it seems a perfect place to introduce our first guest, Sarah, a senior staff scientist who works in the human genetics group here at the Wellcome Sanger Institute. Welcome, Sarah. It's lovely to have you with us. Now, to start off, tell us a little bit about what you do. Tell us, what is it that you are working on? What do you do?
Thank you. It's great to be here. Well, we're primarily interested in the rates of de novo mutations. So those are the mutations that we see in children that we don't see in the parents. And we're also interested in the factors that influence changes and differences in those rates between people. And as we've been moving on and we're able to sequence more and more genomes, now we're interested in how those mutations manifest themselves in rare disease in particular. And so people who have rare diseases that are caused by mutations that have occurred when they were de novo mutations that have occurred between their parents and themselves, if that makes sense.
And you've mentioned this word, and I know you sort of briefly explained it. Can you just break apart this word de novo mutations in terms of and also an opportunity, as we've started to do this introduction, is there anything that we mentioned in our intro that you'd like to add towards or slightly nuance in the terms of the area that you work in?
Yeah, so I thought it was a really great introduction. And most people think of de novo mutations as mutations that we can't see in the genomic sequencing of the parents and that we can see in the genomic sequence of the child. But obviously, as you alluded to in your introduction, this is happening all the time. So generally what that means is mutations that are present at high level or not in the parents of the blood, in the parents of the child, and at high level in the child. And so it's the difference between the two that we're interested in. But yes, the mutations are a continuous continuous process and happening all the time. And so really we're measuring what just what we can see at a point in time. And so for humans, most people will have 60 to 70 differences across their entire genome between themselves and their parents. But as you alluded to in introduction, only 1 or 2 of those will fall into coding sequences. So only 1 or 2 of those will have any effect on your body or who you are. But most of the time, they don't do anything damaging. As you say, they're what we would call benign. So nothing particularly is happening with those. You're passing them safely on to the next generation.
And so you said this word, and I hope when we get into our second guest, we might be able to unpick the slight differences between the two fields. But you've got 60 to 70. As Jack said, we have 3.2 billion letters. 60 to 70 seems really, really, really small. So what is it that is causing these, that 60 to 70 to occur? What is actually happening there?
Well, so we're still in the process of understanding that. And over the process of your germline, so your embryonic development to the point where you have children of your own, you are, as you say, constantly mutating, but at different speeds and at different rates. And so in actual fact, we know that 2/3 of the 60 or 70 de novo mutations you have will probably have derived from the paternal germline. That means they'll have come from your dad. And that is because your dad's sperm is constantly remaking itself over the course of their life. So as your father gets older, you will accrue more generations. So your sibling is likely to have, if they're younger than you, less mutations than you. If they're older than you, slightly more mutations than you, de novo mutations that will have come from their father. And so in terms of the mother, it tends to come from, as you say, damage or kind of over time, damage that's accrued that's not been repaired efficiently. So there's kind of these two processes going on all the time in the cell. And that's what we're seeing. But the germline's really well protected. And if it wasn't, we would be seeing hundreds and thousands of mutations kind of between, because the generation time in humans is 30 years. So obviously that's a lot of cell divisions, a lot of replications that are occurring. So actually we're really well protected.
Yeah, okay, that's really interesting. Jack, I saw your face. Is there something you wanted to come in on there?
No, I've not got a question. I just think it's kind of amazing that, you said that word, so protected. This huge code, if I was having to type out 3.2 billion letters, I'd certainly make a lot more than 60 or 70 errors. I suppose it's just really telling how good those, you said germline, for anyone who isn't studying biology. So that means we're talking about the sex cells, those sperm and those eggs, just how protected they are, how good they are at maintaining that DNA through time.
Yeah, absolutely. So we did a study a couple of years ago where we looked at 20,000 families and looked at the de novo mutations in all of those individuals and found that very few had outside that number of 60 to 70 mutations. And, it's actually very unusual. It was a handful of families that had a greater than expected number of de novo mutations. So it's a very protected biological process.
Yeah. And you mentioned that maybe only 1 or 2 of those mutations might end up in the code and therefore then have some form of an effect. Can you just speak a little bit about, what that therefore means and why it's so important to understand this process in terms of, because you're looking at the developmental sort of stages, aren't you?
Yes, so the majority of mutations will fall in what we call non-coding parts of the genome. So as far as we're aware, they have very little effect. So recently we have found some non-coding genes, some mutations outside of what we thought was the coding region that cause disease. But the majority of the ones that we see in our disease cohorts, children with rare disease, they are loss of function de novo mutations. So they're not seen in the parents because they cause a severe disease. We see one in the child in a particular gene that causes this specific disease, and that's all you need. You need one loss of function mutation in that protein in that gene, and then that gene no longer functions, and so that causes a specific developmental disease.
Okay, and tell us a little bit about, tell us how you go about you mentioned a study that you did with 20,000 families. How is it that how do you study something like this where it's, you're looking at such small mutation sort of rates here, as you say, really highly protected? How do you go about getting all of the data to be able to even look at some of this? Yeah, really,
Yeah.
really important information.
Well, so actually we did one of the first studies where we established what the number was, the number of mutations that you see between one generation and the next. And that first study involved a lot of work because it wasn't far after the human genome. But over time, because sequencing technologies have evolved so fast, and now we're able to sequence genomes kind of in a day. The DDD study, which was our flagship study for children with disease, used exome sequencing. So that just sequenced the small part of the genome that we knew to be coding at the time across a mother, father, and a child, and sometimes a sibling if the sibling was also affected. The kind of disease genomes that we're working with at the moment, particularly through the NHS, who are now doing genome sequencing as standard for patients that are incoming, that's across the whole genome. So that's the whole 3 billion bases, so we can look at the whole all of the mutations rather than just the coding mutations to see if anything outside the known genome, the known bits that are important, have an effect. So that's quite exciting.
And you just mentioned the DDD study, that's Deciphering Developmental Disorders. Is that correct? Yeah. Okay. We'll make sure to share a link to that sort of studies and stuff there. Jack mentioned that obviously mutations can, there are just, random, but there are also environmental factors that can play a part in the sort of area that you're looking in terms of de novo mutations. Do environmental factors play a part here as well, or is it where do environmental factors come into play here as well?
So we haven't found a huge role for environmental factors because obviously that was something we were very interested in for a number of years. So the families that we looked at who had many more mutations in the human families we looked at who had many more mutations than we expected, some of them had damage to a DNA repair gene, which means that when damage does occur, that they can't repair it as efficiently as other people. And there were a very small number, 2 or 3, who might have had chemotherapy when they were young, which affected their germline. But other than that, we didn't find any huge influence from smoking, or in actual fact, the most relevant one is the parental age in terms of how many mutations that you will accrue. So if you like, that's the most mutagenic. In the past, we did also do a study looking at the effect of radiation on mice, because obviously we don't want to look at humans, but that was driven by a concern for people that had been involved in things like Chernobyl. And what we found actually was that, again, there was not a huge burden of additional mutations driven by radiation exposure. So we found a slight elevation of a particular class of mutation, but in general, there wasn't a lot of evidence. And since then, since that study was published a few years ago, it's been replicated in humans who were involved in Chernobyl and other kind of similar incidents, and found the same thing, actually, that there doesn't seem to be a heritable effect from that. So far, no big evidence that there are very strong influences from the environment that are controlling our de novo mutation rates.
So I think this is fascinating, and this feels like a really useful point to bring in our second guest now. But just to summarize where we've got to there, Sarah's working with de novo mutations. So these are mutations that have occurred in a child that we're not seeing in either of the parents, something new happening in the creation of that embryo and the early growth of that embryo. Quite a low mutation rate, not very much change that we're seeing there. Enough to be interested, but not loads, and not really impacted by environmental factors. Okay, I want to bring in our second guest now because they're going to introduce a different kind of mutation. So our second guest is Federico. He's also a senior staff scientist, but this time working in cancer, aging, and somatic mutation at the Wellcome Sanger Institute. Welcome, Federico. It's lovely to have you here with us. I'd like to start, just like Sarah, by asking you to tell us more about what is it that you are working on.
Okay. Yeah, I think it's very good combination to have Sarah and me because the way germline and the rest of the body mutate is very strikingly different. So basically, I don't work with the germline. I work on mutation in the rest of the cells of the body, which are the majority. We are talking about tens of trillions of cells, each one with their own genome. And then all of them accumulate lots of mutations during life. So on average, a cell by midlife, each cell carries like 1,000, 2,000 mutations. And that's just coming from the internal metabolic biochemistry, which endogenous mutation naturally occurring. If you go to tissues like lung in smokers or skin exposed to UV light from the sun, then it's many, many more mutations because this cause many mutations. So we are full of mutations. You can imagine that any base, any one of these letters in the genome is mutated in many cells because there are so many cells and each one carries thousands of mutations. So we know that cancer is a disease caused by mutation. So there's a big interest in understanding this, but we mainly study healthy tissue. So we go to healthy tissue and analyze these mutations and, and look for mutations that when they happen, they make one cell grow better or faster than the neighbor cells. So they are always competing. So we are trying to disentangle how cells regulate each other and how some cells proliferate more. We know that in our bodies there are lots of these cells proliferating more, but that doesn't mean it's going to turn into cancer because the system is quite robust and you need like several parts of the system to be broken by mutation to be able to develop a tumor. So yeah, basically that's our interest. Also, our work requires a lot of technological investigation because it's quite hard to identify these mutations from standard sequencing data. So
Yeah. Great. Thank you, Federico. So you're doing something quite different here. You're not concentrating on these germline mutations, the things that are happening in that early stage when a new child is born and that we see in these sex cells, the sperm and the egg, and how they interact in that early stage of life. You're looking at all of the other cells, quite a big job by the sounds of things. And in particular, you said you're looking at healthy tissues. Now, before we get into those mutations, we heard that was a higher mutation rate in the thousands and much more with environmental factors. I just want to dig into I suppose where I began in my introduction with the group that you work in, because I think some people might be a bit surprised. I said that you work in cancer, aging, and somatic mutation. These feel like quite different things, those first two. But let's just unpack the first word, the last word in that. Somatic mutation is probably a word most of our viewers haven't come across before. What does somatic mean?
Somatic, so when we talk about our bodies, there's a clear distinction very early since the development of the zygote. Is the germline lineage and the somatic lineage. The germline lineage will give rise to the sperm and the eggs, and all the rest is known as the soma. So when we say somatic mutation, we mean mutations that happen in our cells but are not inherited in the next generations.
So I think this is an important distinction for people, isn't it? Is that there are mutations which can be passed down, and so that's where we see inherited conditions or something that flares up in someone's life that might be a risk for future generations. But what you're studying here are things that won't be passed on. They're in all of the cells of our bodies that aren't involved in reproduction.
Yeah. And that's quite interesting because you can understand that the mutations that you pass are much more dangerous. And we find that the germline cells are much more well protected against mutation. We don't understand really why, but it teaches us that there are ways to deal with mutations more efficiently than in standard cells.
Yeah. So yes, that is really interesting. I like that you frame that in terms of the kind of risk, I suppose, to the species or to the family of things in those germline cells. So just again to highlight that difference, you said most somatic cells, it's perhaps 1,000 or a few thousand mutations that we see within that cell's lifetime. Within the germline cells, the sperm, the egg cells, we're only seeing 60. Do you have an idea of why that is? I know you said that it sounds like there isn't a consensus of why that is. Do you have a working theory?
No, I think we don't know. Obviously, it provides an advantage, so it makes a lot of sense. It may involve more production of genes that repair the DNA. It may involve a different internal metabolism and biochemistry that produces less reactive compounds that may damage the DNA. Yeah, so I cannot give you an answer. I think we don't know yet.
That's good. I really like it when we have these things in our videos where the scientists have to say we don't know, because that means that you out there, one of you watching this, needs to come along and help us solve these questions. But great, yes, some ideas there that it might be the DNA is working harder to keep itself repaired. It might be that the cell somehow is operating in a way that creates less damage. That's some great ideas. The name of the group as well is Somatic Mutation Involves Cancer and Aging. Tell us why those two things are connected.
Well, we are learning still, is very early stages, but we are learning how mutations are related to aging, and we are learning, and not only to cancer, but to other diseases. Yes, for example, there's this interesting study done by a colleague in different mammal species. There are mammals like mouse that live from 2 years to humans, which live up to 90, 100 years. And there's all entire spectrum of lifespans in mammals. And in this study, what they found is that there was a very strong correlation between the lifespan and the rate of mutation their cells were accumulating, in such a way that by the end of their lifetime, regardless it was 2 years or 100 years, cells from the colon specifically in a mouse or in a human had a very similar number of mutations in the thousands. So there are different interpretations. We don't yet know, but this could suggest that mutations have a role in aging, and species that live longer are trying to keep down the mutation rate. Yeah, but basically, I think it is still to be disentangled.
I want to bring you back in at this point, Sarah, because I want us to try and tackle. We've heard, I suppose, how much the DNA is mutating so far, but of course, the title of this video is "How does it mutate?" I started right at the top here talking about DNA as a code, and when we study the DNA, we use these letters to signify it. But of course, that's not what's happening in the body. In the body, these letters represent chemicals. So how does it happen that one chemical becomes a different chemical in our DNA code? Sarah, I'll come to you first on this.
Well, so what we think happens with de novo mutations is that there's during replication, which is when the cell is dividing, it just makes an imperfect copy of itself at that particular, because obviously it's copying itself. So that's one way that it changes. But another way is also if it's damaged and somehow it needs to be repaired. So the part of the chemicals you say needs to be put back on, that the repair enzyme comes along, and occasionally it doesn't repair everyone well. So one times in, I don't know, 100,000, it will put the wrong piece of the chemical puzzle back on again. And then that's maintained next time the cell divides into another. So there's these constant kind of processes of like change going on in all our cells, I guess.
Can I jump in?
Yeah.
Regarding the copying of DNA, this has been thought to influence a lot the accumulation of mutations, but we have some recent results that suggest that it's not that important, and it agrees with experiments that have shown that the enzyme, the protein that copies the DNA, is extremely accurate. It makes very, very few mistakes. And we've compared the accumulation of mutations in cells that go through many division cycles, so they have to copy their DNA multiple times, to others that haven't gone through all these cycles, we barely notice any difference. So I think there's strong evidence that the copying of DNA is not that important for the creation of new mutations, although this may need to be corroborated. So I think it's more the other thing that Sarah said, that DNA becomes damaged and this brings in repair mechanisms. Some repair mechanisms are able to fix the error correctly, but other mechanisms are mutagenic. They alter the code.
What I think is really amazing that you've sort of mentioned there, Federico, is that, as you just said, recent studies are starting to indicate that it might not be, and starting to answer some of those questions that you were both exploring in your different avenues. But again, for those that are watching, about how much is still being explored by your different groups, and trying to unpick those mysteries, I think is always so fascinating when we get two amazing guests like yourselves here today. Federico, I wanted to go back a little bit to that thing that you mentioned to Jack about just how many cells that are in our human body that fall under this sort of umbrella of somatic cells, somatic opportunity for somatic mutation. I guess it's that question that I think you tried to allude to a little bit, but is where, as researchers and scientists, how do you decide where to start investigating across so many of those cells where there's potentially so many mutations happening? Where do you begin those investigations? Where does that start, I guess?
Yeah, that's a very good question. Sometimes it's down to technical reasons, or, for example, we originally learned the most just studying cancer, because cancer are groups of cells that all share the same mutation, so that makes the mutations more easily detectable. But then when we went to healthy tissue, we tried to look for groups of cells, but now they are much smaller, that may share those mutations. And there are these kind of structures, for example, in the colon, there are colonic crypts. There are small, like, invaginations that the cells there all come from a single cell, and they are continually being replenished, so they share the mutation. So I have colleagues that go with the laser microscope and dissect these microscopic structures, and with a special protocol send them for sequencing. So I mean, the ability of finding these groups of cells has been a key factor in deciding what to study or where to start. And recently, there are other technologies we've been involved in developing that allow you to study basically now any tissue, because you no longer require to find these groups of cells sharing mutations. You can just have all mixed. And the trick in these new technologies is so if when you are looking for groups of cells that share mutations, that gives you the advantage that you are going to see the mutation multiple times, so you can distinguish from noise. Now that we don't require that, the trick is to read the same DNA molecules multiple times, so you can remove the noise. That's what is allowing us to go for any tissue.
Okay, so lots of potential now with the development of also new technologies with sort of the fields. I just want to shift back to you, Sarah, in the sense of something that we haven't mentioned, and it might be interesting to just unpick a little bit. But obviously, your area and your group is under the human genetics. But I just wanted to Federico mentioned in sort of somatic mutations comparing to other species. I just wondered if there was any, in terms of de novo mutations, if there was any comparisons with other species that is interesting to make in terms of from a human perspective, or any interesting things that exist with other species that is, yeah, sort of quite fascinating in terms of differences.
Oh, definitely. I think so. We've done mouse de novo studies because those were the easiest ones for us to look at a few years back, because they have a short generation time. So that means, 2 years between generations, and they have lots of offspring, lots of children. So we can sequence all of those and look at that. So we looked at the rate, timing, and the spectra, which is the difference of the type of mutations between mice and humans, and found that mice had between 13 and 20 de novo mutations per child, if you like, which seems like quite a lot given that their lifespan's only 2 years compared to 60 in humans, when ours is like 30 or longer. But that could be because if you're a mouse, you have 11 offspring per litter, so you have many offspring over your lifespan, which kind of goes back to Fede, what Fede was talking about with terms of our mutation rates are very highly constrained, because if we had that many, we don't have that many children over our lifespan anymore. So if we had as many as mice did, then we would all be very mutated. I know that there are other studies going on, particularly in the cancer, because there is a synergy between these two programs. I know that these guys are also looking at de novo mutations in primates and other species, because the other thing we found is that there are some mutations that have what we would call a selective advantage in sperm, which means over the course of your lifespan, if you're a man, your kind of pool of de novo mutations that you pass on to the next, or you are more likely to pass a particular mutation in particular genes onto the next generation. So the older you become, the smaller your pool of progenitor cells to pass on to the next generation becomes. And there are certain types of dynamics in that pool that particular genes are kind of overrepresented, if you like. So I know that there's work going on to look at what's the driver behind that, because those do, in some cases, they also mean that you're more likely to pass, your children are more likely to have a rare disease as you get older, because these genes share similarities, these kind of driver genes, as we call them, share similarities between themselves, and some of them do cause rare disease as well.
I think what's just really, yeah, I think that's something that it would just be great to pause on, and then I'll hand back to Jack. But I guess is that you know, if for anyone that doesn't know, the Wellcome Sanger Institute, based at the Wellcome Genome campus, lots of staff, 2,000 staff, lots of different groups. What I found is always amazing is just when we come together in that synergy between, and hearing about, you both are looking at different things, going quite specific sometimes with the different research, but then when it comes together in the collective sort of, as you mentioned, synergy between the two programs to try and basically unpick the mysteries of human DNA and cells, and how we age, how we've developed, and in terms of from an early stage to a later stage. I find it's Fascinating. I always learn so much when in these calls. Jack, yeah, back to you.
Yeah, really, really interesting. I wondered whether we could pull out a little bit and get a wider perspective on where the field is in terms of looking at both de novo and somatic mutations. Federico, perhaps we come to you first. What is it? What do you think are going to be the big questions, mysteries that we don't know now that we're going to start to solve over the next few years? And perhaps, if I might add to that, what do you think the impact will be outside of academia? Outside of the wonderful people like the two of you who work in this as scientists and researchers.
I think key questions. So we've done many studies with small groups of donors because this was very complex, but we've started to see that the patterns of somatic mutation are different between people, not only due to external environmental factors like smoking or exposure to UV light, but some things that seem to be intrinsic. And I think going for larger and larger cohorts is going to teach us a lot of to spot this pattern and patterns of different patterns and find out the underlying cause, genetic cause, to understand better how cells regulate their proliferation, how DNA is repaired. And that's like the basic biology perspective, but in terms applied to society and health, we are finding already hints of links to disease in the mutations, how the liver mutates might be related to how the pancreas is behaving, and in autoimmune diseases there are also some hints, and I think there's a lot of room for exploiting that and to treat diseases and understand them better. But yeah, I think it's exciting, but very early stages.
Yeah, it's so much within genomics there's a newness to this science that I think is really exciting. I mean, we've been doing it for a while now, but I think the technology has changed so quickly that the scale of what's possible has changed very quickly too. And we come up with this almost every video of we're really just beginning on this. And that's what I kind of heard there, Federico, is that yeah, there's lots of new vistas to uncover. Yeah. Sarah, I wonder if I put the same question to you. What in the field of de novo mutations don't we know now that you think we will know over the course of the next few years?
Actually, that's quite exciting, because one thing we haven't discussed is we've covered the rate and how many we'd expect to see, but we haven't looked at the function. We haven't talked about the function of those that we see at all. And at the moment, something like 40% of the patients that are being recruited to the NHS to go into their their genetic medicine service have a rare disease, and about 25% of them have a diagnosis. There are many undiagnosed people with rare disease. And at the moment, we can't tell whether the de novo mutations we see are causative of that disease easily. And so one of the things we'll be doing in the next few years is developing different methods to basically look and catalogue what the effect of each mutation has in particular genes. And that will be of clinical and therapeutic use for people who are able to say, well, that de novo mutation is causing that disease, and we may be able to target it with this therapy. So really, it's about massively accelerating diagnosis and then treatment of rare disease and common disease as well. But obviously, rare disease is what we're studying.
Fantastic. Well, thank you. That feels like quite an uplifting moment, I think, to wrap up. We've had a really fantastic discussion so far today. I know there's lots still left to talk about. You're completely right, Sarah. Of course, we're going to need to have you both back for a second video, which is not just how does DNA mutate, but what happens when it does. I suppose that feels to me like a pretty good place to go next. But thank you to both of you, to Sarah and to Federico, for joining us for this episode, which is how does DNA mutate.
Thank you.
Thank you.
Oh, it's Sam. I can see that you're speaking, but the sound is not coming through. I don't know what you're saying.
Oh dear, technology, eh? Just at the end, it's failed me. I just was trying to say thank you to everybody watching. But yeah, thank you for joining us. Do make sure you check out our other videos on our YouTube. And next month we have another Genomics Lite. So after this video, you'll be notified of the next session. So click notify me, and you'll get a notification when we're going live for the next session. But yeah, it's been wonderful. Thank you, everybody, and see you next month.